Scientific research and medical testing generate an alarming amount of waste, especially plastic and hazardous chemicals. The global biomedical sector alone produces over 5.5 million tons of plastic waste every year. Unlike household waste, lab waste is often contaminated with hazardous substances, making recycling difficult and increasing its environmental impact. Chemical waste is another major concern. Many solvents, acids, and reagents used in experiments are toxic to ecosystems. Heavy metals in lab reagents like mercury and cadmium persist in the environment and accumulate in living organisms. This contamination affects water sources and soil quality, which disrupts entire food chains. Single-use plastics like pipettes, vials, and reagent bottles are frequently used and discarded after a single experiment, playing a significant role in waste generation. Biological labs also contribute heavily to waste. Used agar plates, culture media, and genetically modified organisms can pose biosafety risks if not properly managed. The National Institute for Health reported one facility, Novartis Pharmaceutical Laboratory, produced nearly 15 tons of animal waste in 1 year. Single-use plastics in these labs, especially pipette tips, microcentrifuge tubes, and petri dishes, are used in large quantities and contribute significantly to landfill overflow and ocean pollution. In medical facilities, estimates of hazardous waste production suggest up to 11 kilograms per hospital bed per day. These supplies, when not properly managed, remain in the environment for decades, further harming ecosystems. While disposal regulations exist, enforcement varies across institutions, leading to inconsistent environmental protections. As someone studying biochemistry, I've spent a lot of time in chemistry and biology teaching labs. Something that's always stuck out to me is how much waste we go through in every lab session. Over the semesters, I've started to notice just how much waste gets generated in these settings. In chemistry labs especially, solvents and other liquid reagents are used and then poured into designated waste containers. While I know they're handled according to safety protocols, I've always wondered what actually happens to that waste once it leaves the lab.
Studying biology, I too have questioned lab waste practices. The use of animals for dissections and testings are vital components for education and research. But how can we be more sustainable without limiting our knowledge capacity?
This summer, I'll be working on research to synthesize a new antibiotic, which will involve using even more chemicals and generating waste. This has made me realize just how much waste is produced in research settings, which feels almost contradictory. On one hand, we're advancing science and tackling important issues, but on the other, we're creating a growing amount of waste without really acknowledging the environmental cost. This has consequences not just for students and researchers like us, but for all of society and nature alike. The contradiction and widespread impact prompted us to dive deeper into this topic.
There is no simple fix, but researchers and institutions are already finding ways to address the issue.
One promising approach is using biodegradable lab plastics made from plant-based materials, which break down more easily than regular plastics.
Additionally, using less toxic and more biodegradable chemicals can reduce environmental impacts.
Chlorine is commonly used as the primary chemical reagent for disinfection. Liquid and powdered forms of diluted chlorine are easily accessible and provide a gentler footprint. However, even more impactful alternatives like vapors, steams, and UV light have shown to deliver identical cleaning effects.
Recycling is often considered the best option for reducing waste. Once cleaned, plastics can be processed into smaller pieces and reformed into new products through both open and closed loops. Reusing plastics is both cheaper and more sustainable for the environment. The Department of Environmental Health and Safety at the University of St. Thomas is responsible for the disposal of hazardous waste. In 2023, the department reported that the majority of universal wastes were directed towards recycling stations and programs, many of which can be found around campus. Other institutions have implemented on-site chemical recycling systems where solvents are purified and reused instead of being thrown away.
Another key step is reducing waste at the source. One attainable method is switching to non-contact supplies. For example, infrared thermometers eliminate the need for millions of single-use plastics every year. Another strategy is improving management of purchasing and inventory control. Refusing or limiting supplies with short shelf life can cut down on unnecessary environmental contamination. As college students, practical solutions like these are essential. With lab waste becoming a bigger problem, every small effort matters. Using biodegradable materials, recycling, and adopting simple, attainable practices bring us closer to a more sustainable future. With more awareness and better access to programs and resources, we can all contribute to protecting our planet.